If we take a cross-section of the kidney, there are two main parts, the outer cortex and the inner medulla. If we zoom in, there are millions of tiny tubes called nephrons, which go from the outer cortex down into the medulla and back out into the cortex again. Nephrons filter out harmful substances in the blood so that we can excrete them into the urine. Each nephron is made up of the glomerulus, or a tiny clump of capillaries, where blood filtration begins. These capillaries have very thin walls and they act a bit like a coffee filter. Red blood cells and proteins that are large stay in the capillaries, whereas blood plasma and smaller particles get filtered out. This filtrate, called tubular fluid, collects in a cup-shaped structure containing the glomerulus called the Bowman's capsule. Together, the glomerulus and the Bowman's capsule make the renal corpuscle. The Bowman's capsule is connected to the renal tubule, which has a few segments. First, there's the proximal convoluted tubule. Then there's the U-shaped loop of Henle, with a thin descending limb, a thin ascending limb, and a thick ascending limb. And finally, there's the distal convoluted tubule, which empties into the collecting duct, which collects the urine. Zooming in on the U-shaped loop of Henle, it's lined by epithelial cells. On one side is the apical surface, which faces the tubular lumen, and on the other side is the basolateral surface, which faces the interstitium between the tubule and the peritubular capillaries. The peritubular capillaries run alongside the nephron, and the capillaries that run along the loop of Henle are called vasa recta. Solutes and water that are reabsorbed into the interstitium go into the vasa recta and then re-enter the circulation. Now, when the tubular fluid leaves the proximal tubule, it has an osmolarity of around 300 milliosmoles per litre, which is the same as the osmolarity of the interstitial fluid around the tubule. The tubular fluid mainly consists of water, sodium, potassium, chloride, calcium and urea. When it travels down the descending limb, the osmolarity in the medullary interstitium increases rapidly from 300 milliosmoles per litre at the top to 1200 milliosmoles per litre at the bottom, where the loop bends. Most of that osmolarity in the interstitium is due to large quantities of sodium as well as some urea. Now, the squamous epithelial cells that line the thin descending loop have a lot of aquaporin proteins on the apical and basolateral sides. These aquaporin proteins are channels that only allow water to pass through. So in the thin descending limb, water diffuses from the lumen into the tubular cells and from the tubular cells into the interstitial fluid, where the water gets reabsorbed into the vasa rectas and then returned to the circulation. The epithelial cells have very few channels for solutes, so solutes don't leave the tubular fluid in significant quantities. Since water leaves but solutes remain in the tubular fluid, it quickly becomes concentrated, and at the end of the descending limb, the tubular fluid has an osmolarity of 1200 milliosmoles per litre, just like the interstitial fluid. Now, the thin ascending limb is also made up of squamous epithelial cells. But these cells don't have any aquaporin proteins, so the thin ascending limb is impermeable to water. Instead, the thin ascending limb has lots of sodium and chloride channels at the apical and basolateral surfaces of the cells. And these sodium and chloride channels allow those ions to diffuse from the tubular fluid into the epithelial cells, and then from the epithelial cells into the interstitium, all of it down their concentration gradient. Since water remains in the tubule, but solutes move out, as we travel up the thin ascending loop, the interstitial fluid starts decreasing in osmolarity, reaching around 600 milliosmoles per litre at the top of the thin ascending loop. After the thin ascending limb, there's the thick ascending limb, which is made up of cuboidal epithelium. These cells are much bigger than the squamous cells, so that's what makes this portion of the loop of Henle thick. These cells also don't contain aquaporins, so the thick ascending limb is impermeable to water. Epithelial cells along the thick ascending limb have sodium-potassium chloride cotransporters on the apical surface, and they shuttle one sodium into the cell down its concentration gradient, and that powers the movement of one potassium and two chlorides into the cell as well. Think of it a bit like a revolving door, where sodium is the guy doing all the pushing, and then the potassium and the two chlorides just kind of follow him in. On the basolateral surface of the tubule cell, a sodium-potassium ATPase uses ATP to pump 3 sodium ions into the interstitial fluid in exchange for letting 2 potassium ions into the cell. Finally, both chloride and potassium move from the cell into the interstitial fluid passively, through their own channels down their concentration gradients. Now, the volume in the tubular fluid is much lower than the volume in the interstitium. But if sodium is pumped into the interstitial fluid over and over, then the interstitial fluid will become more concentrated, and we end up with this highly concentrated interstitium in the medulla. And since solutes keep moving out as we travel further up the thick ascending loop, the tubular fluid starts decreasing in osmolarity, reaching around 325 milliosmoles per litre at the top of the thick ascending loop. The process of creating this gradient is called countercurrent multiplication. It uses ATP to build up the concentration gradient, but in return we get to reabsorb water into the interstitial fluid for free, since it can diffuse passively via osmosis. Now, importantly, the thick ascending loop is the target of loop diuretics. Specifically, they attach to the chloride bonding site of the sodium-potassium chloride cotransporter and inhibit the transport protein. Diuretics are medications that help the body to lose water by inhibiting the reabsorption of sodium. Water follows sodium, so if more sodium is excreted into the urine, more water is lost in the urine as well. Alright, so as a quick recap, the loop of Henle has three main parts: the thin descending limb, which is where water moves out of the tubule through aquaporin proteins, the thin ascending limb, which is where sodium and chloride move out of the tubule through channel proteins, and the thick ascending limb, which is where sodium, potassium and chloride move out of the tubule through a carrier protein and channel proteins.